Welcome back for the last session of today. It is my very great pleasure to introduce Thomas Flatt, who's going to give the first talk of this session. I've been following Thomas's work for a while, especially because he was interested at some point in senescence and evolution of aging, which I don't think is the topic of today's talk. But Thomas is professor of evolutionary biology, and he's also head of the Department of Biology at the University of Fribourg. And regarding his different steps, I'm not going to go too back in the past, but he did his PhD in Fribourg, and then several postdocs, especially funded by the Swiss NSF. And then in 2017, he became SNSF professor in Lausanne before he moved back to Fribourg. So another interesting point is that he co-leads with Josefa Gonzalez, who's around here, the Drosophila Population Genomics Consortium. And today he's going to talk about an ancestral balanced inversion polymorphism that confers global adaptation.
Thank you very much, Anne, and thank you, Virginie and Abdu, for having me. It's a great pleasure to be here, and I hope you guys all had enough coffee during the break. So we will continue to talk about chromosomal inversions, also in a fly, but in fruit flies, not in seaweed flies. But you will see some similarities in these two systems. So Drosophila melanogaster, which is the system I'm mainly working with, is a long-standing genetic model for climate adaptation and for understanding spatial adaptation. And this species is a subtropical, tropical insect, as we have heard this morning. It's of sub-Saharan African origin, and it originated in the area of Zimbabwe, Zambia, in southern Central Africa. And as far as we can date this, I mean, there's some uncertainties about this, it migrated out of Africa and underwent major population bottlenecks, something like 10 to 20,000 years ago. So subsequently, the species migrated out of Africa and then colonized other climate zones, including temperate climates, and is now a cosmopolitan. It's also a human commensal. It likes ethanol. It eats bacteria and yeast that is growing on rotting fruit. And so now we have it everywhere around the globe, including in our kitchens. And so that makes it a very good model, both for demography, but also for spatial adaptation. So this history of spatial adaptation colonization actually has led in this species to the formation of many latitudinal clines. And we see these latitudinal clines on pretty much every continent and subcontinent where we look. And so these clines can be seen both at the genome-wide level with genomics, with sequencing, but also for markers like microsatellite structural variants that we will talk about today, but also many phenotypes, including many phenotypes that are clearly fitness-related. Things like body size and fertility and so forth. And so here I'm showing you the example for body size. So here we have the example of the North American East Coast, where we have an East Coastal cline that spans populations in Maine, Vermont, up to Canada, and down to southern Florida. And we see a matching clinal gradient here in the southern hemisphere along the Australian East Coast, running from tropical Queensland down to places like New South Wales and Tasmania. So what we see here for body size is that high latitude populations are genetically larger as compared to low latitude populations. And we see the same matching pattern on the southern hemisphere, and this is actually also true for other continents and areas. More importantly, we see this pattern not only for body size, but also for other fitness-related traits. So there's parallel latitudinal clines at both the genotypic and phenotypic level. Now, in our previous work, we have found that much of this clinal behavior, both genetically and phenotypically, is actually driven or underpinned by a large chromosomal inversion, which is called IN3R pain. Here, you see it here on this polythene cytological preparation in terms of this heterozygote loop in the heterokaryotype. And so this is a large inversion. It's about 8 megabases large. It's paracentric, and it sits on the right arm of the 3rd chromosome. So this inversion encompasses something like 1,200 genes. And we know that it explains clinality because if we sort of do genome-wide analyses of clinal variation, for example, along the North American cline or along the parallel Australian East Coastal cline, we see that something like 80 to 90% of clinally varying SNP outliers map to this inversion. Of course, they're not going to be independent because linkage in this inversion is high. We also know that this inversion is of sub-Saharan African origin. It's of monophyletic origin. And it has been dated to be something on the order of 130,000 years old, maybe older than that, and we will get back to that. So this is roughly 2 NE generations or more. The interesting feature of this inversion is that it's globally common. There's like 4 globally common inversion polymorphisms in this species. This is one of them. And if we look on global average, it has an average frequency of something like 15%, which is not huge, but it's quite considerable, because in many places, this inversion actually does not exist. It's absent in terms of polymorphism. On each continent, as I'm going to show you on the next slide, we see latitudinal frequency clines that are very pronounced. We also know from our own work that when we isolate inverted chromosomes or karyotypes and standard non-inverted karyotypes and we phenotype them, that this inversion polymorphism affects many fitness-related traits. So we can think of it similar to Claire's example as being a life history supergene. So it affects developmental time, it affects viability, how big the flies are, affects things like fertility, their adult lifespan, and cold shock resistance. So it's really a sort of a life history supergene. So here are the latitudinal clines around the globe. This is based on a meta-analysis of 34 data sets spanning over 50 years of data, including genomic data. And so we see this inversion to be at low frequency or at 0% frequency in temperate high latitude populations. So the base of the arrows here for North America and for Europe and so forth, or for Japan and the Indian subcontinent. And it's always, as far as we can tell, at a sort of an intermediate frequency in subtropical, tropical, low latitude populations. So for example, here in southern Florida, or here in Queensland, where it reaches frequencies of 30 to 60%. As far as we can tell, it doesn't go to fixation, which is peculiar, and we see this pattern on pretty much every continent. If we look in the existing African data, we don't see pronounced clinality, which is also interesting. And there's new data from South America through the Drosophila collaboration that we're part of that finds also a similar upside-down cline as we observe in Australia. So the clinal pattern reversed on the southern hemisphere. So what I want to talk about today is a population genomic and population genetic analysis of this inversion polymorphism across 4 continents to look at spatial patterns. So this is a paper that is currently in press at MBE, and this effort was spearheaded by 2 talented former postdocs, Martin Kapun, who is now at Vienna, and Ezra Durmas-Mitchell, who is now a senior postdoc at the University of Southern Denmark. And this work is in collaboration with Paul Schmidt at UPenn and Tad Kawecki at Lausanne. So here are the population genomic samples that we have analyzed, both published and unpublished, all phased sequencing data so that we can look at haplotypes. And so in total, we looked at data from 485 strains on 4 continents: Africa, Europe, North America, and then we also had some published data from another group from Australia. These data from Australia, however, are based on rat sequencing data, so they're like, in terms of resolution, not that amazing. So for some of the analysis I'm going to show you, we had to leave these data out. A special focus of interest was for us to look at the ancestral range population samples from Zambia, because that's where this inversion presumably originated. So in this paper, we sought to address 3 fundamental questions. The first one being whether patterns of variability and divergence that are associated with this polymorphism, whether they're consistent with recent or long-term balancing selection. And this is an interesting question because in Drosophila melanogaster, people have supposed that most chromosomal inversion polymorphisms are probably pretty young and might not be subject to long-term balancing selection. The problem, however, is that This is slightly at odds with the fact that these inversions have a quite high intermediate frequency, including in Africa. So we wanted to look at that. Then secondly, we wanted to see, by looking at genetic data, whether this inversion is in some sense locally adapted. So Dobzhansky had this idea for the inversions that he studied in subobscura, that maybe the same chromosomal rearrangement, the same chromosomal inversion might capture different locally adapted alleles in different populations. And we wanted to see whether that's the case, or whether adaptation conferred by this inversion is based on the same sort of haplotype. So the question then would be whether the inversion confers perhaps some species-wide adaptation, and whether it might have captured a single adaptive haplotype in its ancestral range prior to migrating out of Africa. So we looked at population genetic patterns. I'm going to show you in a minute. And so the theoretical interpretation of these results was greatly helped by regular conversations with Brian Charsworth. So Brian has a new paper out on the archive where he's derived new coalescent predictions for neutral genetic variants that are associated with inversion polymorphisms. And he also performed some calculations based on our data. So what does actually theory tell us about, in terms of predictions for a long-term balanced inversion polymorphism? So an inversion polymorphism subject to balancing selection is expected to create population subdivision into inverted and standard chromosomes. It sort of causes a partial selective sweep that eliminates part of the variability. It is then expected to establish itself at some intermediate frequency. And then over time, very, very slowly, this system should sort of approach mutation drift recombination equilibrium with respect to neutral variants. That is, it will over time, given a sufficient time, experience recombination, gene flux, as Claire has mentioned, which will homogenize the variability between the two arrangements. And it will, of course, also accumulate new mutations that are lost or fixed by drift. So let's unpack this a little bit more. So at such an equilibrium, what sort of patterns might we expect in population genetic data? So one thing that we would expect is linkage disequilibrium between targets of selection and neutral sites that are linked to the targets of selection. We would also expect LD among the neutral sites. We should see divergence between haplotypes that carry different alleles at the targets of selection. And we might also see enhanced variability around the target of selection in the population as a whole. So how would this look like for an inversion? So here we have two cartoons of inversions where the stippled lines here denote the inversion breakpoints. And so for the scenario of balancing selection, we might expect, for example, that the targets of selection, they could be sitting at the breakpoints, where also recombination or gene flux is very strongly suppressed. And if this inversion is sufficiently old, towards the center of the inversion, we know that there's considerable gene flux over time. And so here, a variability or divergence would be more homogenized, but we would still see some elevated divergence even there. An alternative scenario could be that there might also be targets of selection in the center of the inversion, away from the breakpoints, with all these peaks being in linkage disequilibrium, and with these peaks in the center being maintained over time despite homogenizing gene flux, sort of carving out this suspension bridge pattern with additional peaks. So we were sort of interested in comparing our data to these predictions. So the first thing I want to show you is our patterns of nucleotide variability, pi. And so this here is for the 4 continents. And what we can see here at the breakpoints is that there's reduced variability at the breakpoints, as we would sort of expect, because there at the breakpoints, recombination is supposed to be strongly suppressed. And then we see increased variability towards the center. And this might also be consistent with the fact that this inversion has some substantial age. So homogenizing gene flux might have happened in the center of the inversion. So there's an estimated mean flux rate for this system here of about 10 to the minus 5. So if this inversion is indeed sufficiently old, then we expect that variability will have homogenized. And eyeballing the pattern, you see that actually the pattern for standard chromosomes, shown as the gray line, and the colored lines are the inverted chromosomes, that these patterns of variability are rather congruent and similar. However, if we zoom into the breakpoint regions and look at nucleotide variability there, we see some subtle differences. So here the light gray dots show you nucleotide variability for the standard arrangement and the black dots for the inverted arrangements. And you can already appreciate here for 3 continents that pi tends to be higher for standard as compared to inverted inside the inverted region. We also see here, comparing the distal and the proximal breakpoints, that pi is higher for the inverted arrangement at the distal as compared to the proximal breakpoint. And so that means that the patterns of variability at the distal and proximal breakpoints are discordant. And so this, in fact, the only good way to explain that is that the suppression of gene flux at the breakpoints is not complete. So these patterns are consistent with an equilibrium model of balancing selection with recombination. And under such a model, we would expect that the presence of a newly arisen inversion creates population subdivision, and it will increase diversity due to the accumulation of new mutations that distinguish the inverted and standard chromosomes. So that will result in the coalescent time being somewhat increased for standard alleles due to the partial population subdivision, while the coalescent time will be reduced for the inverted alleles. So Brian performed some calculations based on our empirical patterns, and that seems to be compatible with such a model. So this inversion is not super old, but it can't be too young either, so it seems to represent a balanced polymorphism. So what about patterns of divergence between karyotypes? So this is shown here, again for the 4 continents, and you clearly see if we calculate an FST-like statistic for divergence, that we see very strong peaks of divergence at the breakpoints. And then you see elevated also values of FAT or FST away from the breakpoints. And again, the values we get here are sort of consistent with long-term balancing selection. Now, interestingly, if we go to Europe, North America, and Australia, so the derived populations, we see the same peaks at the breakpoints, but we also see massive center peaks in the middle. So this sort of smells of this cartoon that I showed you before, where we have center peaks away from the breakpoints, where over time gene flux should homogenize divergence between the arrangements, where perhaps these center peaks might be maintained by selection. But that's a speculation because, of course, these are derived populations and they have undergone a bottleneck. So we cannot exclude that these central peaks here are due to a bottleneck. On the other hand, given enough time, these peaks should have become homogenized. And so it's a matter of timing. But so overall, these patterns are also consistent with long-term balancing selection. So similar patterns can be seen when we look at linkage disequilibrium. In fact, the patterns for linkage disequilibrium sort of match the patterns for divergence. And so here, this is just an example for the population sample from Florida, where when we look at the pool of inverted chromosomes or when we look at the pool of standard chromosomes, there's no LD whatsoever. That makes sense because for these 2 karyotypes within a karyotypic class, there's free gene flux. There's no reduction in recombination. But when we look at the pool of inverted standard chromosomes, all the LD is there, and that reflects also the major divergence we see between these arrangements. So we see very strong LD between the breakpoints and also center peaks in the middle. And so this is, again, sort of suggestive of long-term balancing selection. It could potentially indicate multi-locus balancing selection. So what about if we just now look at divergence between inverted chromosomes themselves? So Dobzhansky had this idea, and similarly Kirkpatrick and Barton in 2006, that perhaps the same chromosomal rearrangement will float around and sort of capture different adaptive alleles in different populations. And so when you go to a different population, you might find slightly different adaptive alleles. But when we look at FST, between continents for these inverted chromosomes, we actually see very low differentiation. And the same is true for standard chromosomes. So here for these inverted chromosomes, we sort of argue there is not much evidence for locally adaptive alleles. In fact, when we quantify allele sharing of inversion-associated candidate genes and SNPs, we see very significant allele sharing across all continents. And so based on these data, we sort of argue that this inversion probably has captured, when it first arose in Africa, one ancestral adaptive haplotype and sort of exported this around the world. So we believe that this inversion might actually be conferring somewhat global or species-wide adaptation that has led to the parallel formation of clinal gradients around the world, but it's always the sort of same allelic content that drive these patterns. So this is what we find here somewhat inconsistent with these local adaptation models that were postulated by Dobzhansky and Kirkpatrick and Barton. So can we actually sort of prove this? This is really hard. But so Mark Kirkpatrick in a conversation gave us this idea. And so Mark said, like, okay, so if this inversion arose in Africa and captured an adaptive haplotype there, maybe you still see some of the alleles that got captured by the inversion among standard chromosomes, that they're still segregating there. And so that's what we tried to look at in this analysis. And so what we did here is that we said, like, okay, let's define inversion-specific, highly diagnostic alleles based on the sample from Florida with a FAT value greater than 0.9. And so these are highly diagnostic. They're nearly fixed for the inverted chromosomes. And let's look at their frequencies in North America, Europe, and Zambia. So here are the Florida-specific alleles. Of course, they're going to be fixed on inverted chromosomes in Florida. That's how we have defined them. Standard chromosomes in North America are fixed for the alternative allele, so that all checks out. So let's look at their behavior in Europe. The same pattern is true. We see the American Florida inversion-specific alleles to be fixed among Portuguese inverted chromosomes, but to be absent or to be fixed for the alternative alleles for the standard chromosomes. What about Zambia, the ancestral range population? So here, interestingly, we see that these inversion-specific alleles are polymorphic and segregating among both inverted and standard chromosomes. And they actually produce a very interesting frequency differential here. So they're at 75% frequency here and roughly 20% frequency here in the right direction. So these alleles could potentially be alleles that were captured by the inversion. And now when you look at derived populations, they're polarized in this way. But of course, that's no proof, and we're currently further looking into the nature of these alleles. Most of them sit at the breakpoints, but some of them also sit in the center of the inversion. Okay, so this was a rapid gallop through a complicated subject, but so in conclusion, IN3R pain is a globally common balanced polymorphism. It's a major determinant of latitudinal clines around the world. It affects multiple fitness components that themselves are clinally varying. And when you look at patterns of variability and divergence, it suggests that this might indeed represent a long-term balanced polymorphism. And so this inversion might indeed have captured a single adaptive haplotype in its ancestral African range prior to becoming cosmopolitan, and then sort of assorted itself in a parallel fashion along these latitudinal clines. The big elephant in the room is that we don't fully understand what sort of balancing selection is at play. And so one thing that we're very interested in, which connects to both Claire's and Dimitri's talk, is fluctuating balancing selection. We don't see clinality in Africa, but we do know that in derived populations around the world, this inversion not only shows latitudinal clines, but also seasonal frequency fluctuations that are repeatable. And in fact, in the paper that Dimitri showed, in the Machado et al. Paper, this was one of the inversions that is enriched for seasonal signals. So we're quite interested in studying this further. And so with this, I would like to acknowledge my collaborators, current group members, funding, and thank you very much for your attention.